All right, so I would like to talk a little bit about the intersection of serotonin and autism. I won't go heavily into that background because I want to get into experiments that, as researchers here, I think more interested. Really what I want to do is primarily introduce the main target of my presentation, which is that green blob there at the end of that cartooned presynaptic terminal, and that's the serotonin transporter. It acts like a little nano vacuum to clear serotonin out of the cleft after release and maintain levels at an appropriate level for the kind of neuromodulation that serotonin is supposed to be delivering. Most people know about this protein because it's a target of SSRI class antidepressants like Prozac. We cloned the gene that encodes this protein many years ago now, and its sister. Did I do that? No? Okay. I'm going to keep going because I want to keep you guys on time. So, and we cloned various members of this gene family. We've been studying their regulation for a number of years. And I spoke a little bit yesterday about the fact that we had found early on in our studies, once we had gene probes, that this gene was expressed very highly in the embryo of a mouse. Essentially, at about day 9, day 10, it comes on quite actively, as do many serotonin receptors in their expression in the brain. And this moved part of our lab away from the study of mood disorders per se to juvenile onset disorders or even embryonic onset disorders that might involve serotonin signaling. So back in 2005, and that's a long time ago now, using a linkage-based design to identify DNA that was being selectively transmitted to affected children with autism in collaboration with Jim Sutcliffe's lab at Vanderbilt, we identified a peak that overlay the serotonin transporter locus and about 100 other genes. And it motivated us to go and take a look at what kind of genetic variation in the transporter might be there. We were interested in that because of that developmental picture that I was describing, but also traits of hyperserotonemia that had been described for 2 decades before that, looking at individuals with autism who had a suspicious serotonergic marker of very unclear, and still to this day somewhat mysterious benefit for us as scientists in understanding autism. We're a protein lab. We work on the transporter protein, so going after coding variation, even if it was rare, was for us really the goal. And if you went out in a community sample of 300 families, which we used in this study, and looked for coding variation in the transporter, you'd find very, very little, if anything, because most of these are rare. We found 5 coding variants in that sweep, and all of them that we identified showed a gain of function. You can see here at the bottom the human serotonin transporter in transfected cells, and then all these other variants showing elevated transport activity for serotonin. And you would see this if you looked in lymphoblasts that came from those subjects. So it wasn't just a transfected cell issue. And this was very suspicious. Why would all of them be going in the same direction, or what did it tell us? Well, fortunately, in another part of my lab, because we were focused on this protein in the basic science arena, we were uncovering pathways that normally stimulate the activity of the transporter. And one of the main pathways we found was one that could be initiated with the application of inflammatory cytokines, specifically here showing IL-1 beta and TNF alpha in either a dose or time-dependent manner, stimulating the transport activity in a serotonergic cell line, RN46A, which are neuroblastoma derived from the raphe of rats. So we had worked out this pathway and in vitro, and then we injected IL-1 beta into the hippocampus next to an electrode that could sense the clearance of that serotonin, a carbon fiber, and the rate of that clearance can be read out on that carbon fiber as the time it takes to sweep away 80% of the serotonin from that electrode. And what you can see in the bottom right corner there is that the time to clear serotonin when you have IL-1 beta injected is much lower. So the clearance is faster in concert with high levels of uptake that we saw in vitro. So a lot of other studies I'm not going to go through brought to our attention a pathway in serotonergic neurons by which IL-1 beta, acting through the IL-1 receptor, signals through P38 MAP kinase to put the transporter into a catalytically active state. I don't have time to talk about the molecular dynamics of that. I'd love to offline. But for now, I just want to place that pathway in your head. IL-1 receptor, P38 MAP kinase, to an activated receptor. In this case, one that's already surface resident. It doesn't have to traffic. It can just move into a more active conformation and give you enhanced uptake for a short period of time. Now, this was quite interesting in relation to autism because by that time, studies were emerging in postmortem brain looking at transcriptomes and different pathways that were altered in the brains of individuals with autism. And this is just one study out of Karoli Mernix and Tony Persico's group. And what you can see in terms of the pathways that were significantly changed relative to normally developing individuals were, in fact, IL-1 receptor stimulated signaling pathways, P38 MAP kinase stimulated signaling pathways. So we thought that maybe perhaps there was a merger here of this activated state of the transporter into risk for autism, whether that was coming genetically or coming about through an environmental insult, perhaps via inflammation through the IL-1 pathway. So our model became that we had two states of the serotonin transporter. The one on the left with its smaller black arrow transports a certain amount of serotonin. It has an allele over there in red at the bottom called Gly56. That's the most common of the variants that we studied, and we decided to focus on it. That's the normal transporter, and you get the normal regulation when the IL-1 receptor is stimulated. P38 MAPK gets activated, puts the transporter, what we call the SERT star state, or the more active state, enhances clearance, eventually relaxing back down once phosphatases and whatnot get in the game and shut this process down, returning it back to the normal SERT state. But in the Allo56 case, what you have is the transporter is sitting in that state all the time. And we could show this was refractory to P38 MAPK activation, suggesting that it is in fact an end step in that pathway of P38 going to SERT activation. Now this became even more meaningful once we took a look at where IL-1 receptors are expressed in the brain, and this is in the brain of a mouse. And what I'm showing you on the left is a coronal section through the mouse midbrain, stained for serotonin. You could also use TPH2, the enzyme that makes serotonin, and you'd see the same pattern. You'd see a beautiful sort of butterfly shape, wing shape of serotonin neurons in this dorsal raphe area and also continuing down into the median raphe. And what you see when you look at where the IL-1 receptors are located, you see a very rich expression of these receptors. In this particular plane of the section, you see that these dorsal and dorsolateral wings of the raphe are very intensely expressing. Now if you go throughout the brain and you look at where IL-1 receptors are located, you see two principal areas where it's incredibly intense. The dentate granule cells of the hippocampus and the dorsal raphe serotonin neurons. Okay? So this is very conspicuous. It's not distributed all over the place. You see it certainly in endothelial cells and blood vessels, but you just don't see neuronal populations under normal conditions expressing a high level. These neurons do. What did they do there? So this is slice recordings by Hideki Omoto and my group looking at the firing of dorsal raphe neuron slices that we can detect the serotonin neurons because of a transgene. They express, we patch them, inject current to get them firing at a rate that's physiological, apply IL-1 beta, and you can see a very nice inhibition of excitability. You see firing shut down, you see hyperpolarization of the resting membrane potential, and that reduction that you see here can be blocked with co-application of IL-1 receptor antagonist peptide. When we do a voltage step across different potentials that we're holding and look at that effect and subtract it, what we find is that what we're inducing with IL-1 beta is a characteristic inwardly rectifying ion channel. The reversal potential suggests that it's an inwardly rectifying potassium channel that's being activated. We haven't really narrowed this down and gone after this at a molecular level, but I think for right now, I think it's good evidence that in fact the serotonin neurons can sense this inflammatory cytokine at the cell body level by turning the firing of those neurons down. That means less release at the terminals, and they turn up the transport activity, contributing to an overall dampened serotonergic state. So we wanted to test this model in vivo, and one of the ways, and we also wanted to know, how often is it that you get pure IL-1 beta go up in an inflammatory event? Usually you get some kind of complex mixture of cytokine elevations when you have an inflammatory event. So let's try something a little more complicated. So we did this amperometry approach with animals, unanesthetized animals. These are anesthetized. These are anesthetized, and we inject LPS peripherally. And as you can see, as a function of time, here in the individual traces and here in the time plot, the clearance rate increases, meaning the time to clear 80% goes down as a function of time after injecting LPS. It hangs around for about an hour with this low dose that we inject. And you can see the clearance rates going down, down, down. That means it's faster, faster, faster to clear serotonin. So serotonin neurons in vivo and the transporter in vivo are sensing the presence of this inflammatory event that just happened in the periphery. If we knock out the IL-1 receptor in those serotonin neurons and do a biochemical preparation to look at the transport activity in the terminal fields, you can see that when we give LPS, we get a nice increase, and that's gone if you knock out the IL-1 receptors in the serotonin neurons. Now, recently, we have adopted an approach. It's now, we're using light sheet microscopy and whole brain imaging. These are from traditional sections, looking at neuronal activation in the dorsal raphe after that injection of LPS. We're using CFOS staining to mark activated cells. And what we're quantifying here on the left would be the wild type given LPS. So this is saline. You give LPS, you see a drop in the number of CFOS positive cells consistent with in vivo inhibition. If you knock out the IL-1 receptor on this side of the graph, sorry it didn't get labeled well, what you see no change. And again, indicating that those serotonin neurons are responding to peripheral LPS. They're changing the number of neurons that are excited during that period of time where we're activating CFOS. And you can see this in a male mouse in the lateral wings. You can see it in the dorsal part, ventral, and in the interfascicular region. The female's a little bit different. In the three ladder regions, we see essentially the same thing. The lateral wings don't respond like the male does. And on some other data, if I had time to show you, you would see that the female also recruits the median raphe to this kind of response. The male does not show it. So there is some heterogeneity within the raphe serotonin neurons by sex. It needs to be worked out more, and of course we're interested in a sex bias disorder, so I think that's going to provide some interesting information moving forward. Now, I want to relate those data. Those are all in either wild-type animals or animals we've engineered to manipulate the IL-1 receptor. I want to go back to those mutants that I showed you that we had identified, because we made a mouse that carries that most common variant, the one I depicted as a glyph or alanine substitution, very subtle change, but it turns out it's in a really critical part of the cytoplasmic N-terminus, one that bears a lot of regulation. We made this mouse. We went to look and see what are its phenotypes, and what we found immediately to our delight was hyperserotonemia showing up in the blood. This is the first time that biomarker had ever been established in a mouse model, let alone one that had some connection to an autism phenotype. You can see the ALA56, again with this amperometric approach and recording in the hippocampus, showed this increase in clearance rate, this more steep drop of serotonin when you inject a fixed amount near the probe. We found that the protein, the serotonin transporter protein, is hyperphosphorylated, and that hyperphosphorylation occurs through a P38 MAPK pathway. I'm not going to go through the pharmacology, but it was incredible to see that in vivo, that this subtle mutation must be occurring in some kind of important regulatory domain linked to P38 MAPK. Now that's very important because that opens up pharmacology for us to see if what we can do to shut down this process in vivo. A few more phenotypes. The firing of the, that is also reduced relative to the wild type slices. Let's go here and let me now hit play, see if I can get past it. Okay, so behavior, of course, matters more than anything. We did a number of behavioral tests, but I'm just going to show you one. This is the tube test, typically used for social dominance examination, but can also just be used to encourage social interaction and determine whether the animal finds it aversive and study components of it. If you come out nose first when you meet two mice meet in the middle, you're considered a winner. That's carryover from dominance literature field. And what you see from here is that the Allo56 mice are significantly have a lower probability of coming out a winner. They back out of this interaction. All right, so what can we do about it? Well, we had that pathway, the P38 MAPK pathway, and about this time, a reagent was developed by my colleague that I had known for many years, Marty Watterson, up at Northwestern. And he developed a CNS penetrant P38 alpha MAPK specific, there are 4 isoforms, alpha specific inhibitor. We gave this drug to this animal, one dose, didn't do a dose titration. We used some of his data that he was using to study neural inflammation. We gave it once, didn't do anything, nothing changed. We gave it once a day for 5 days, and now what you saw, sorry. Now what you're going to see is that that clearance rate now normalizes. It now becomes the rate of clearance. In fact, I think I might just stay for the sake of time. I just don't want to be going back and forth on that. Now you can see that these two curves overlap. Clearance rate for the wild type and the mutant treated with this P38 MAPK inhibitor are the same. Similarly, remember what I told you? Here are the mutants now against, it looks like they're actually elevated, but they're not in many trials, they're just normalized. They have a normal probability of a social interaction in that assay. They also, well, I won't get into that. Now, to the title of this presentation, what is going on with these Allo56 mice that is leading to this hyperphosphorylation? They're clearing out serotonin more actively. So there's this tonic level of depletion, let's say. They're firing less and they're clearing more. What does that generate? Well, we thought about it from the literature. It looks like there's good evidence that serotonin actually quiets microglia. There are several serotonin receptors whose activation will prevent them from becoming activated. So we went and looked in the midbrain at a number of inflammatory cytokines by qPCR, and you can see IL-1 beta levels, IL-6, and IL-10 elevated in terms of expression in the Allo56 mice. And if I can play this one, let's hope I can get through this. Didn't do this on my computer. Anyway, so there's evidence that there are a number of factors that are secreted, some by neurons and some by other kinds of cells, that will keep microglia in a quiescent state. One of them is serotonin. And our belief then is that with this reduced availability of serotonin, we have this tonically elevated state of inflammation. It's not huge. It's not like you have a trauma to the head, We're not talking about 10-fold elevations or even 3-fold elevations. We're talking about somewhere between a 50% and a 2-fold elevation. But we believe that that's substantial enough to set these neurons down a path where they are probably part of the way down toward a full-blown shutdown. And now a small event can trigger a much larger effect. It could on its own, if they had this mutation, do it. We don't think that that's a causal mutation. We think it's just one of those background risk factors. But this has illuminated a biology that we'd like to study more and think that perhaps this is one of those routes by which inflammation targets a specific behavioral driver, one that can control aggressive behavior, social interactions, competitive behavior, and these things were seen in our mice. All right, with that, I'm just going to thank the people that have worked in my lab, a lot of them over the years, and of course the funding of the Brain Foundation in small ways on this project that were very important in keeping continuity in the project. So I'll stop. 2 minutes up. Maybe I could take a question or two. Yes?
Can you comment on the ALA56 association studies in the human population outside of ASD? What other indications do you think?
No, they're really, all right, so this variant, the ALA56 variant, is present at about 1% of US population, right? So that's pretty common for variants in our field in these transporters. Very few have the coding variation at that level. That still presents an issue with respect to the numbers of individuals who have been in association studies, and almost none of them have genotyped for that. So we've had to turn around and go backwards into larger genomic data sets and try to see what traits it maps onto. A lot of those data sets don't have individuals with autism. They don't have individuals with psychiatric diagnoses. So we're kind of at a loss there. My guess is that it's one of those complex factors. It's buffered by a lot of other things, and so unless you get the right group and the right background, you might not see anything. So we just haven't had any success with a global association. We were able to do a modest association study in those 320 families we looked at, but we're not talking about a lot of variants in there.
One thing I never understood is how an increased activation of the transporter causes an increase in blood. Do you have any?
Yeah, so most of the, almost all of the blood serotonin is in the platelet, it's trapped. Unless that happens, it's degraded. So the transporter that's on the platelet takes it up from the circulation, it stabilizes it essentially in the platelet. So if you have more transport activity, you're going to get a larger serotonin pool circulating, and that's what we're picking up in our mice, right? If you want to be careful, I don't think that SERT for the hyperserotonemic individuals is the sole, maybe even not the major way to get there. It points to a pathway, though, by which anything that stabilizes the uptake, storage, reduces metabolism, changes any gross properties of the platelets, that will happen, right? So that's what I think is happening in those mice. All right, why don't I stop, because I think we have an excellent talk coming right after me.
Thank you so much, Dr. Blakely, and thank you for adjusting with the change in schedule. And I really want to apologize that projector failed.